thank you for having me here. Thank you for this award. Thank you for the warm introduction. So today I will be talking to you about one of the major findings during my PhD about the Duffy antigen receptor for chemokines or DARC. But before talking about that, I would like to give you a brief introduction on GPCR. GPCRs represent the largest class of cell surface receptors. They comprise of more than 800 different members in the human genome, and they can perceive a wide array of chemically divergent stimuli ranging from odorants, photons, gustatory molecules to ions, proteins, peptides, and even lipids, steroids, and nucleotides. And in response, they play an important role in regulating almost all of our major physiological systems. Interestingly, despite comprising of such a large number of members, GPCRs are characterized by an overall conserved architecture. They have 7 membrane spanning helices, which are connected on the extracellular and intracellular side by several loops. So the extracellular loops along with the N-terminus of the receptor constitutes the extracellular domain, and this mediates the binding of the receptor to the ligand. Now once a ligand binds to the receptor, this induces certain conformational changes in it, which has to be relayed onward from the extracellular to the intracellular side, and this is done by the transmembrane domain. And finally, on the intracellular side, the intracellular loops along with the C-terminus of the receptor make up the intracellular domain, which modulates the binding of the receptor to its downstream transducers. Now, GPCRs represent a major drug target with 40% of all commercially available FDA-approved drugs targeting GPCRs. And these are just a few examples of some of the molecules currently available in the market that target this class of receptors. However, the entire scope of drug discovery is yet wide open because, as you can see from the schematic over here, the number of receptors against which established targets are available is quite few compared to the total number of receptors. So to put it very simply, GPCRs have 2 primary transducers, G proteins and beta-arrestins. And if it responds to binding of a ligand, a receptor signals through both G protein and beta-arrestin, then those receptors are known as the classical or the canonical GPCRs. Work in the last decade have identified a subgroup of receptors which do not couple to G protein but signal specifically only through beta-arrestin, and these are known as the arrestin-coupled receptors. Now, in this context, DARC or the Duffy antigen receptor for chemokines represents a very interesting receptor downstream to which G protein and beta-arrestin signaling has not really been comprehensively studied. So, if we take a look at the expression pattern of DARC, then it is found to be expressed on a variety of different kinds of tissues, particularly the surface of erythrocytes and the endothelium of postcapillary venules. And why I specifically single out these 2 sites of expression is because DARC expressed in these regions is targeted by several pathogens. So for instance, Staphylococcus aureus secrete these bicomponent leukotoxins that recognize DARC expressed on the surface of endothelial cells, and this recognition allows it to bind to DARC, which allows it to form a complex which then inserts itself into the membrane, forming a pore. DARC is also recognized by Plasmodium vivax. So P. vivax merozoites express this protein PVDBP on their surface, which recognizes DARC expressed on the surface of erythrocytes. And this allows the merozoites to come into close proximity to the erythrocyte to form a junction and subsequently gain entry inside the erythrocyte. Now, DARC is classified as an atypical chemokine receptor. It binds to several chemokines belonging to both CC and CXC, and in my today's talk, I'll talk about our findings with one of these chemokines, CCL7. So to begin with, I started out with measuring G protein signaling. So I measured signaling downstream to the 3 major G protein subtypes, G alpha I, signaling through which leads to a decrease in cytosolic cyclic AMP levels, G alpha S, signaling through which leads to an increase in cytosolic cyclic AMP levels, and G alpha Q, signaling through which leads to an increase in calcium ions. And in all 3 cases, we do not observe any response downstream of DARC following stimulation with CCL7. We next measured beta-arrestin recruitment using a protein complementation-based assay, and therein also, in case of the positive control, we observe a dose-dependent increase in beta-arrestins, physical interaction with the receptor, but in case of DARC, we did not observe any interaction. And we wanted to be sure about this lack of interaction, so for this we carried out an orthologous approach. So when we express YFP tag beta-arrestin in cells, in absence of any stimulation, it is widely distributed in the cytoplasm. And in case of the positive control, as you can see, At initial time points following stimulation, it translocates to the membrane and later gets endocytosed. But in case of DART, we do not observe any change in its distribution pattern. And so we concluded that DART does not signal through either G protein or beta-arrestin. So we were interested in understanding then how does DART actually signal in the cells, and for that we undertook a phosphoproteomics assay. So we generated stable cell lines expressing DART. We had two conditions, one which we stimulate the cells with CCL7 and one in which we did not. And then we enriched for peptides and identified them. So we observed that several peptides undergo an increase and decrease in their phosphorylation status following stimulation with CCL7 downstream to DART, and that these are involved in regulating different molecular functions and biological processes, one of them being the endocytic or the transcytotic process. Additionally, we also undertook an interactome analysis, and our aim was to identify those proteins which are physically interacting with DART. So for this, we performed a flag co-immunoprecipitation. So how does this work? So any protein that was physically interacting with DART, we chemically cross-link the two, and then we pull down the entire complex using the flag tag present on the N-terminus of the receptor. And we observed that several proteins indeed undergo a change in their interaction with DART, and these proteins are involved in regulating different molecular functions and biological processes. And comparison of our phosphoproteomics and interactome analysis identified 66 common hits, which essentially highlights the robustness of the system. Independently, using string analysis, we also identified CD82 as one of the interacting partners of DART. So CD82 is expressed on the surface of primary tumor cells, and interaction of CD82 with DART expressed on the surface of endothelial cells inhibits extravasation of these tumor cells and also upregulates P21-mediated senescence in these cells. So we wanted to confirm the interaction of DART and CD82 using our assay system and observe that indeed, if we co-express CD82 and DART and pull down DART, then we get a co-immunoprecipitation of CD82 as well. And this interaction is unaltered upon stimulation with CCL7. So to better understand how CCL7 physically interacts with DART, we determined the structure of this complex using cryo-EM. And our structure provided us with two very interesting observations. First is this superficial mode of binding that is exhibited by DART. So typically, binding of chemokines to chemokine receptors involves the engagement of this orthosteric pocket on the receptor, which in case of DART we observe is completely empty. And the second observation is the dramatically shortened length of transmembrane helices 5 and 6. Now what these short TM5 and TM6 lead to is that they preclude the formation of a cavity on the cytosolic side of the receptor. Now why is this cavity important? So when we compare the structure of DART with that of another canonical GPCR C5AR1 bound to G protein, or another receptor beta-1, adrenergic receptor bound to beta-arrestin, we observe that in both cases, this cytosolic cavity is actually where the transducer is docking into the receptor and binding to it. And since DART does not form the cytosolic cavity, we hypothesize that that is perhaps why it cannot physically couple to either G protein or beta-arrestin. So to summarize, this study provided us with insights into the molecular activation mechanism and ligand binding pocket of DART. It provided the structural basis of lack of canonical transducer coupling and allowed the identification of novel potential signaling transducers. So I would like to thank my PI, Professor Schukla, for his guidance during this project, our collaborators, Vladimir Korkov, for assistance with cryo-EM, and my current PI, and all past and present lab members. Thank you.